And now we would like to hear from Ilvana, so we'll turn it over to you.
Thank you, Linda. Just share my screen. Oh, sorry. Can everyone see that?
Yes, thank you very much.
Okay, so I will be discussing how we can use computational modeling of brain activity to inspect on more fine-grained deficits during auditory oddball tasks along the psychosis continuum. So this is specifically focusing on EEG. So as scientists, we use models at their very basis to make sense of complex set of interactions and relationships using simple rules. And psychiatry's earliest models attempted to explain the psyche based on psychoanalytic theories on the influence of childhood experiences and defense mechanisms. We have gone far since then, especially owing to non-invasive neuroimaging and more advancements in understanding behavior and cognition. Computational psychiatry allows us to build mathematical models relevant to psychiatric diseases. In computational psychiatry, a data-driven approach is to use machine learning methods to classify illness and predict treatment outcomes and selection. But in this talk, I will focus on a theory-driven approach in computational psychiatry as a way to gauge the underlying mechanisms of symptoms and behavior. Here, we use computational modeling on neuroimaging data as a way to bridge between alterations at the neurophysiological level and neuronal dynamics. So we collected 3 groups of people, including inpatients at a psychiatric ward, so schizophrenia spectrum inpatients and also non-psychotic inpatients that had disorders such as depression and anxiety. And we also had non-clinical controls who might also be part of a clinical high-risk group. In this study, we gauged the psychosis continuum and risk for psychosis using CAPE questionnaire. So this questionnaire assesses psychotic-like symptoms, also in the general population. And what we found was, not surprisingly, our non-clinical population scored the lowest. The non-psychotic inpatients were intermediate, and the schizophrenia spectrum group were the highest on this measure. We also used the PANSS interview to assess clinical symptoms such as delusions and hallucinations. So for those who are unfamiliar with auditory oddball tasks, typically these tasks are run during an EEG. And the classic auditory oddball task consists of different probabilities of sounds, and it sounds a bit like this. So people are really good at forming predictions about what they will hear next. And in this particular example, people would form the predictions that the likely sound they will hear next is this regular short sound. However, when this long, rare sound occurs at an unpredictable time point, there is an error in what is predicted, and a prediction error response occurs in the brain. So this prediction error response is computed by subtracting the brain response to these common sounds from the brain response to these deviant, surprising sounds. And it occurs at about 200 milliseconds. It is a negative deflection and is commonly referred to as the mismatch negativity response, or MMN. In our study, we used an auditory oddball task where we could also measure statistical learning by asking participants, which sound did you hear more often? So in this particular block, when they were asked this question, the correct response would be these short sounds at 80%. So they should click here. Any deviation from that point would we referred to as the statistical learning error. So going into the results and just starting off with the standard EEG analyses at the FZ channel, we found that the schizophrenia spectrum group had attenuated MMN, and this deficit seemed to be specific to the schizophrenia spectrum group, as the non-psychotic inpatients did not significantly differ from the non-clinical group. However, we did not find an MMN deficit on the continuum of psychosis, particularly in the non-clinical group. And this may be because MMN or prediction errors are underpinned by a brain network. And attenuation in MMN results from a variety of different disruptions in the connectivity of this network. We can explore the dynamics of this network using computational modeling and therefore find out which particular disruptions may be related to the emergence of psychosis. So we explored the neural dynamics with dynamic causal modeling. DCM uses a neural mass model to describe the dynamics of neurons, specifically the mean activity of an entire neural populations represented by their average firing rate and membrane potentials. This brain network here has robust evidence to be involved in auditory oddball tasks. So we explored the connectivity between the A1 or the primary auditory cortex, the superior temporal gyrus, and the inferior frontal gyrus. We also looked at different hierarchies of connections. So under simplifying assumptions, these bottom-up connections model prediction errors encoded by superficial pyramidal cells. These top-down connections model predictions, and they're encoded by deep pyramidal cells. And these intrinsic connections model the local adaptation of neurons within a particular region. We use parametric empirical Bayes, which is like a Bayesian general linear model, and we explored the modulation of these brain connections by statistical learning, psychotic experiences in our whole sample, and hallucinations and delusions. So what we can see is that with more severe psychotic symptoms, such as hallucinations, there are weaker forward connections from the STG to the IFG, whereas on the reverse, greater top-down connections from IFG to STG was related to more psychotic-like symptoms across the whole population. So we speculate that maybe as psychotic experiences worsen, these very strong predictions lead to less prediction error updating, attenuated MMN, and weaker forward connections. Okay, so what is happening on the other side of the brain? Well, what we found is that people with greater psychotic experiences and statistical learning errors had decreased intrinsic connectivity in the right IFG, or less adaptation. So IFG intrinsic connectivity has recently been found to be weakened by the administration of ketamine, which is known to produce psychotic-like symptoms and also impair NMDA receptor function. Okay, so in summary, attenuated MMN was specific to the schizophrenia spectrum, whereas aberrations in brain connectivity were aligned across the psychosis continuum. So even in the general population that had some risk for psychosis. We found disinhibition of the right IFG may impair statistical learning and increase psychotic experiences. This may be related to NMDA receptor dysfunction, which will be relevant for precision medicine. So for example, inspecting right IFG intrinsic connectivity, we may be able to assess if novel medications are indeed targeting glutamatergic function, and if this does prevent conversion to psychosis. Also, there is an interplay between stronger top-down and weaker bottom-up frontotemporal connectivity along the psychosis continuum. We now need to explore the direction of this interplay with longitudinal data to really understand the psychosis pathophysiology. So from these results, what we can see is the value of looking at more fine-grained deficits at the brain level using computational modeling to really examine what is going on before illness onset and to inform new medication, which would be relevant for AMP-SCIDs. This work has been published in Schizophrenia Bulletin for more details. So I'd like to finish off by thanking Professor Barnaby Nelson for inviting me to present my work here, and my supervisor at Origin, Professor Stephen Wood, and also Prescient and AMP Schizophrenia. And I would like to acknowledge the researchers that were involved in the work I presented here. Thank you very much.
Thank you very much, Ilvana.